Hello, and welcome back to Human Evolution. Today we're going to talk about our mutations because this is why we've been learning about DNA and RNA and proteins. Because when we talk about evolution, evolution occurs because you have these changes in the DNA that change traits. And that doesn't have to be from mutations. It could be from recombination through sexual reproduction, and we'll talk about that later on. But right now what we do want to focus on is this ability of DNA to become different, right? And that is through this process of mutation. And mutations, by the way, are the only source of new genetic material. Okay. Now, when we talk about mutations, typically we're thinking of what can go wrong, but in some cases it can go right. And so the different types of mutations really come in two big categories. You've got gene or point mutations and chromosome mutations. And when you talk about gene or point mutations, there are really two major things that you can do, and then there are variations of this. And then same thing with chromosome mutations. There are a couple of things you can do here, and we're going to talk about each of them. Okay, so let's start with point mutations. These are changes on the small scale, and we're going to start with these because we understand DNA pretty well now. So let's say that we have this wild type, and wild type is an unmutated strand of DNA. So you've got this DNA right here. Okay, and you can see this one is the coding strand. This one is the, sorry, wow, that was crazy. This one is the coding strand. This one is the template strand. Okay, and here is your strand of mRNA. All right, and this is the amino acid chain that is built from this mRNA. Well, point mutations can occur in really two big types, but we're going to divide them into a couple of things. So first of all, you have a substitution. And in a substitution, one of these is changed into something else, right? And so in this case, instead of having a T in the DNA, it got mutated to an A, which means that in the mRNA, you're going to get something else here, right? And so instead of having U, which is complementary to the AAA and matches the TTT, you're going to get UUA. Now that is different, right? That changes this amino acid entirely into a new one. Now this is called a missense mutation, and in a missense mutation, it means that you change an amino acid by changing one base pair. Okay, so you change a nucleotide that gives you a change in a base pair. And there are different versions of this and all this good stuff. And sometimes it matters and sometimes it doesn't. Fortunately, some of the amino acids have similar characteristics, right? And so it doesn't actually change the function of the protein. And if you're very, very lucky, it occurs in this nice little third position here, and it doesn't do anything at all. You actually still end up getting the same amino acid, which was something that we talked about earlier when we talked about wobble positions and stuff like that. Now sometimes when you get this change here, you can see that this changes when you change this G here, right? It changes the template strand to a C, which means that the mRNA gets a G, and that means if you change that here in this case, let's say it's replaced by an A, well that means that the template strand turns into a T, which means that the mRNA is going to turn into an A. Well, UAG is a stop codon. This is a nonsense mutation because it actually stops and doesn't finish translating the chain, right? The mRNA sequence. Okay. Now, other things that you can do, you can pop in an extra one, right? You can see here, here you had the TGG, that's great, and then it was ATA. Well, in this case, we've inserted a thymine, which gives us an additional, right, nucleotide, and that shifts that codon set, because remember the ribosome is reading 3 at a time. Well, now instead of reading ATA, it's going to read TAT and then AGC instead of GCC. Okay, and that changes the rest of the polypeptide. Yeah, you can also delete one, right? And so in this case, right, we've got TTT, and then this one actually deletes one whole codon and one nucleotide. And really, if you delete just 3 nucleotides, it changes the polypeptide, but it doesn't shift anything, right? If you delete or add, you get that frame shift, right? And frame shift means that you actually change the rest because you push off the codon reading, okay? And so this is just an example. Here's a functional protein. Now when you change this one here, you get a different conformation, you get a non-functional protein. And that's going to be really problematic because now that protein can't do whatever it's supposed to do, and that could be a real big problem. Which brings us to the idea that most mutations most mutations really don't seem to make a whole lot of difference anyway in the grand scheme of things. But mutations that do make a difference, they're actually mostly deleterious, which means that they impede the function, or they actually inhibit the function of the protein entirely. It's not really that common that you get something that's beneficial. Okay, so here's your first question, and it is, which of the following would be the best place to have a mutation if you have to have one? The first position of a codon, the third position of a codon, in a region that codes for ribosomal RNA, anywhere's fine, or nowhere ever. Okay, remember going back to that idea of the wobble position, the one that's going to make the least amount of difference is to have a mutation in that third position of the codon. Okay, now where or when are mutations most likely to happen? Well, during replication, right? When you're copying this DNA, sometimes you have mistakes, right? Now, fortunately, you've got proofreading systems and all kinds of repair mechanisms, and that's super duper. But occasionally, it doesn't work out well. And that is going to be happening. So the cell cycle, right, is you have the cell is growing and all that good stuff, and some things are being duplicated. Then the DNA along the way starts to replicate, and you get a whole nother copy of all of the DNA in the cell, and then it checks to see if there are any mistakes, and then cell division begins. This is the cell cycle, okay, and we'll talk about it a little bit more. Cell division is actually called, or the process up to cell division is mitosis, and then the division of the cytoplasm and the plasma membrane is cytokinesis, okay, and we'll come back to that again. But anyway, let's say that this becomes a G, so instead of having instead of having AGT, you would have AGG, and this would change to TCC. Okay, and just to show you, here we go. Anything, by the way, that changes the DNA, that changes the nucleotide base in a DNA strand, is called a mutagen. A lot of times those are chemicals. If you're talking about external factors, sometimes it's just a mistake, right, as you're going through and replicating. But anyway, what happens is you have some sort of altered parent DNA. When it's replicated, one strand is replicated faithfully. The other strand is also replicated faithfully, but it is not the same as the original parent strand, okay? And so you get one normal daughter strand and one altered daughter strand. Well, now, as we go along, that altered daughter strand is going to replicate as well, and you get copy after copy after copy of these mutated DNA strands. Now, the other thing is you can have all kinds of stuff happen, okay? We were talking about substitutions and frame shifts and all that good stuff, but sometimes you can have the nucleotides interact with each other. UV, when you bombard DNA with UV light, like tanning or something like that, it actually causes thymines to bind to themselves. It breaks that bond here between the two nucleotide bases, the thymine and the adenine, and it causes what's called a dimer. And so you get this weird bulge in the dRNA, a kink, if you will. And so this actually has to be repaired, and your body has a great mechanism for repairing it. Invisible light actually helps that, which is great. But if it doesn't get repaired, okay, you have this situation where this bubble will actually cause the replication process to fail, right? And so you can get breaks and mistakes and all kinds of stuff. It can cause really serious DNA damage. Now let's say that something else happens and you get a dimer that forms between something else, right? And so, okay, so now we have this dimer here, right? And we've got replication. Well, this side's going to have normal replication, okay? Well, this side can really kind of put anything you want in there. And so you have this change from CC to TT because you can put anything you want in there, right? And so this side ends up being fine, and this, you could have a little mutation there, okay, because the AA on this side, you know, whatever. Anyway, right, so do you understand here, this has been inserted because there was nothing, because these were attracted to each other, so you could put anything in here while it's being replicated. That's how that happened. Okay, so let's talk about chromosome mutations. Now, the first thing that you need to understand is what a chromosome is, but these are going to be changes on the large scale. So you've got your DNA, your double helix, and it is coiled up around these little proteins called histones, okay, right here. And these are going to, and then here's your histone, right? It's going to coil up around these, right, and coil up around these. And then it's going to coil again, okay, and then it's going to coil again, right? And so you get the helix, which is a kind of a coil, then you get a coil, then you get another coil, then you get another coil, then you get another coil. And this supercoil, right, is going to create these little X-like structures, or at least these little lines, right? This is actually two chromatids together, okay? And what you get in the end is this. All of that DNA is all coiled and coiled and coiled and coiled into this little chromatid right here. And these are, well, they're not identical, but they're the same, right? And so if this is chromosome 1, this is also chromosome 1, and you can see they're connected in the center, right? Here's your chromosome, here's one chromatid, that's an arm, here's the other chromatid. The ends are actually called telomeres, the center is called the centromere, the top short arm is called the P arm, the long arm at the bottom is called the Q arm. And we are going to talk about the centromeres and the telomeres later on because they become very important when we're talking about humans versus the other apes, okay? So just keep in mind this shape. So there are genes all in here, okay? Now, all right, chromosome mutations are where you have changes to that whole structure, okay? So let's say that you start out with this cute little chromosome. It's got genes A, B, C, or segments A, B, C, D, E, F, right? Here's the centromere. Let's say that B just gets deleted. Bye-bye. Okay, so now you've got this big change, and it's shorter. Let's say that you get something that's duplicated, okay? That means that you have a segment that's in there twice. Interestingly enough, by the way, the proteins that are made from these genes will now occur twice as often because you have more copies. Here, with the deletion, they won't occur at all. Now, sometimes these get all kind of twisted up, and that causes breaks and then repairs, and so it's called an inversion. You can see that D and E, right, have switched places with C and B, and they've just switched all the way around. So it's A, B, C, D, E, F. This whole thing is turned, okay? And then sometimes you'll have what's called translocation. You'll have multiple chromosomes, and when they cross over their little arms, they break off and then join the other chromosomes. So you can see here A, B, C, D, E, F, G, H, I, J, K, L, right? Well, in this case, J, K, and L and D, E, and F switched over, and so the genes that used to be on this chromosome are now on this chromosome. And you can just see them again here. Here's duplication. You can see this is doubled. Here's inversion. You can see that it's turned all the way around. Deletion, you take something out. Insertion, you add in something new, like in this case, this moved from here to here. And this, by the way, when you have a segment of a chromosome jumping into another one, this is really interesting, and it can sometimes be caused by things called transposons that will actually go in and excise this area and insert it into another place. And then you have translocation, where two parts of genes, this part and this part, in this case, will actually flip, and this part will move to one chromosome and this part will move to the other, and that's what you saw up here. So deletion, that's this one. Duplication, that's this one. Inversion, that's this one. And translocation is this one. Okay, so the other type of chromosome change that you get are changes in the actual number of chromosomes, okay? And so here are just some examples. There are actually two things. There's aneuploidy, where you have multiples of or deletions of individual chromosomes, and in euploidy you have copies, multiples of the entire genome, right? And so when we start talking about that, this is a really cool thing that's actually done in genetic engineering and stuff like that, biotechnology. Let's say that you have this little haploid or diploid set of chromosomes. Tetraploid means that you have twice the number. So if you have two, diploid, haploid means one. So if you have two copies of it, that's diploid. Most cells are actually diploid, right? If you have 3 copies, that's triploid. If you have 4 copies, that is tetraploid. Now, when this happens in animals, it is very bad. It does not go well most of the time. When it happens in plants, it's great sometimes because plants are very tolerant of chromosomal mutations. And so you can do lots of things with plants. But when you have those really like giant fruits, like the giant grapes that we see in the cafeteria, which are awesome, and those giant strawberries and stuff like that, that is because they have polyploidy. They exhibit polyploidy. They have lots of copies of the genome, and so they essentially have bigger cells. Now, you can also have this SOMI issue, right? This, which is called aneuploidy. And SOMI means you're talking about one chromosome, where ploidy means you're talking about the entire genome. Now, in this case, you'll just have like maybe one chromosome that has too many or too few. If it's supposed to have two, like in this case, you can see all the rest of these have two. This one has three. This is called trisomy 21, by the way, and it's better known as Down syndrome. And it's this extra little chromosome here that causes that entire genetic disease. Okay, and how does that happen? It actually happens when you're making gametes, like eggs and sperm, if they are not distributed, if the chromosomes are not distributed appropriately. Like each of these, if you start out with 4 here, each of these should have 2, right, chromosomes in them, but they didn't split right, so these got 3 and these got 1. So these are not going to give when they join with the other egg or sperm. If you have a sperm, it's going to join with an egg. If you have an egg, it's going to join with a sperm. If the other one is normal, right, then you will either be 1 short or you will have 1 too many. Okay, so in which process would chromosomal mutations be most likely to occur? Replication, transcription, translation, mitosis, or meiosis? Replication is going to be point mutations. That's where you're going to have that. Okay, transcription and translation, they're just going to do whatever they've been given from the DNA. Okay, that's, I mean, you could get some, issues there, and that definitely happens, but not in what we're talking about. Mitosis is where you are you've copied the DNA, and then you are going to split and get 2 new cells. Definitely happens, but it's typically going to be a replication process in that. It doesn't have to be. There can be chromosome issues with mitosis when the cell is dividing. Maybe they don't divide evenly. But meiosis is actually going to be the process that causes the most problems, because it is in meiosis that you actually get crossing over. Now remember, meiosis is the making of gametes, eggs and sperm, and only the female and male germ cells, remember our stem cells, only the female and male germ cells do this. Mitosis is just the replication and division of any cell, okay? So in meiosis, one of the cool things that happens is that when these sister chromatids align and their little, centromeres join, and it's very exciting, their little legs, right, arms, their little arms will cross over, and they will actually normally get these translocations, right? And that's a normal thing. But see, normally you have this even split, and so it's an even trade. It just jumbles up the genes, and that's very exciting for variety, and we'll talk about that when we talk about natural selection. Sometimes it does not go well. It splits and lines up in the wrong place, and that's where you get some of those issues, okay? And then, of course, when you're actually splitting into those final gametes, that's where you can get distribution problems. So you can have problems in the crossing over and problems in distribution, okay? So that's just about what are the types of mutations. So what are the results? Well, normally when we think of mutations, we think of something like this, and this is actually real. It definitely happens. But this is not really, and this does happen to humans as well. But really, when we start talking about mutations, oftentimes what the result is a genetic disease. And these are 3 big ones that are actually caused by just mutations in one gene, okay? And cystic fibrosis is where you have just this single, it's a mutation in both, and you'll understand this more later on when we start talking about genetics and chromosome pairs and alleles and things like that. But anyway, so you have this disorder where you have this mutation in this particular gene, and it causes huge problems for the respiratory system, right? Sickle cell anemia, again, it's in the hemoglobin, right? And so you have this beta globin gene that is mutated, and it causes red blood cells that should look like cute little baskets to look like this, or to look like this sickle shape, okay? And that's a huge problem. And here's the actual mutation right here. And you can see how that changes, okay? These are all hereditary because they're genetic. And then Huntington disease is where you have neurologic issues, right? And that is caused by a mutation in the Huntington gene. Now, sometimes mutations are not all bad. It is mutations that give us the beautiful variety that we see in our own species. We come in all sorts of wonderful phenotypes, and those are all based on mutations. And your ability to even drink milk is a product of mutation, and that's good because milk is good for you, right? Now, here's your next question. All surviving mutations in humans are product of selection. True, false? That is false, actually. Many of the mutations that we see are not about selection. It turns out the ability to drink milk is all about selection because people who can drink milk have higher nutrition, right? Or they don't have higher nutrition, but you have the ability to get more nutrients in this one location, which back in the day when we were actually hunting and gathering and domesticating early on, that's a really good thing, okay? As far as things like eye color and all of these other differences that you see in all of these lovely young ladies, these are mostly about just genetic drift. Populations get isolated, mutations occur, they're not sharing genes because, we spread out really fast and we get fairly far apart pretty quickly. And so some of these mutations are just in these populations, but your eye color doesn't help you environmentally, right? It doesn't help you be more successful. It just is a thing. And so most of these traits are just a thing, yeah? Okay. Now, let's get back to bad mutations, because lots of them are. It is mutations that actually cause cancer, right? And cancer is really cells that have gone out of control. Now, cancer, unlike a lot of other diseases, is not based on a single mutation, okay? You have to have 4, 5, 6, whatever mutations. Most of the time, it's a minimum of 4 mutations, and they have to be in very select genes. There are key genes that these mutations occur in. And in many cancers, we see, like, there are maybe 2 genes that are common to, like, all cancers. Anyway, so you get some mutation and the cell's normal, but it probably bypasses the checkpoints for division, and so the cell can just like divide as much and whenever it wants to, okay? And then they get another mutation, and then it actually like really starts doing that. It goes into overdrive, okay? And then you might have mutations where you have structural changes, and maybe it's something like in your DNA repair mechanism, you get mutations that inactivate that. You may get mutations that inactivate the ability for the cell to actually undergo apoptosis if it's too damaged to continue. And then maybe you get another mutation where the cell is growing out of control, right? And you get these clearly not good cells, right? And then these are the malignant cells. And then, of course, they metastasize and spread to all different sites, and then they call it wreak havoc wherever they go. But cancer is based on mutations. Okay, so the last thing that we want to talk about is the molecular clock, okay? And the molecular clock is the way okay, let's try that again. The molecular clock is us using genes to estimate the time that has passed between the divergence of species, okay? And so basically we're talking about how long ago did something split from a common ancestor, yeah? And mutations occur at a fairly constant rate in related species. And so you can use the number of mutations that have occurred between 2 species. So where are the differences? So here you have species 1 and species 2. Here's the common ancestor. You can see that each one of these has 1 mutation. Well, if mutations in this molecule are occurring every 10 million years, then if you have 1 mutation, you know that it's been 10 million years. After another 10 million years, you should have 2 differences, right? And so really there are actually 4 differences, but it's 2 in each organism, right? 2 in each species. So in this case, you're looking at these guys diverged 20 million years ago, okay? All right, so what molecules do we use? Well, different molecules are going to have different mutation rates. If they have a higher mutation rate, that's great for studying closely related species. The lower rates are better for studying distantly related species. And we really use 2 things. Not so much nuclear DNA, because nuclear DNA is going to be recombined during that meiosis process, and you get half from dad and half from mom. But mitochondrial DNA is unique, right, in that respect that it is from mother to child, mother to child, mother to child. And so mitochondrial DNA will actually track the matrilineal lineage. Now, you can also use Y chromosomal DNA because that is passed down from men, right? Because only men have it. So you can look at the maternal DNA, you can look at the paternal DNA with Y chromosomes. And we'll talk about both of these things a little bit more later on. But mitochondrial DNA, it has a mutation rate that's about 10 times faster than that of nuclear DNA. And so it can show you differences in really closely related species, which is very helpful. Okay. Now, ribosomal RNA, remember that ribosomes have to function very specifically. They do a very specific job, and if they are changed in any way that changes their structure at all, then they will not function. And so they are highly conserved, is what we say. There is a limited number of mutations that can occur that will actually keep those functioning. Okay, so they have a much lower mutation rate than most DNA, and so we can actually use ribosomal RNA to study distantly related species. And that, as a matter of fact, is how we came up with the classifications for animals that we have today. It is in fact based on ribosomal RNA. So when we talk about those prokaryotes and those eukaryotes, and we divided the prokaryotes into archaea and bacteria, all of that categorization was actually based on ribosomal RNA. Okay, so here's your last question. Which molecule would you use to study the relationship between humans and Neanderthals? I hope you have heard of Neanderthals. If you have not, you will shortly in this course. All right, nuclear DNA, mitochondrial DNA, the number of chromosomes, ribosomal RNA, or messenger RNA. Well, we're pretty closely related. I mean, they're close cousins of ours. They just went extinct about, I don't know, what, 30,000 years or so, something like that, 30, 40,000 years. So we would want to use mitochondrial DNA because we're very closely related. Messenger RNA is just, you know, a transcription of nuclear DNA, and we already talked about the problems with nuclear DNA. And so these are right out. Ribosomal RNA, probably going to be really, really similar because we're really closely related. And then the number of chromosomes, that can be helpful for sure. And we'll talk about places where we see that. But it's not really going to be helpful for estimating the time in this case. All right. If you have any questions, please let me know at the beginning of class, and I will talk to you soon. Thanks for listening.